This is gene manipulation part 2. Today we will talk a little bit about the different applications that you can do, and I would like to use some real life examples for this. So we will look at overexpression, a knockout and a knockdown, and we will briefly highlight the CRISPR-Cas system. For example, in this case we have a wild type tobacco plant, and in this case we have a transformed tobacco plant, and in this case it's able to produce astaxanthin. Astaxanthin is a very high valuable product, and therefore it's very nice if plants can make this product in a very easy way. So this is basically, we put a gene that was not previously there, and we put it in the plants and see that it's working. Now, why? Well, the first we have had already, to create a specific product. Another possibility is to increase a product. So for example, we have already a product that is present in plants and we want more of it because the concentration is a little bit low, or we only have positive effects when we have more of it. Or we want to decrease a product in the case that a specific product is not good for us, and it's not good for the growth, or it's limiting in some way. So then we want to decrease this product, or in some ways we want to do some research. And in this one I will give an example. My research is about understanding the quenching mechanism of LECSR. LECSR is very difficult to study in the host organism, so what we want to do is we want to express the LECSR gene into a Arabidopsis thaliana MPQ4 plant. Now the MPQ4 plant is a specific knockout, a PSPS knockout, because it doesn't have any PSPS, but we will get into that later. So how are we going to do that? Well, in the previous video you have seen that first we need to select the gene, then we are going to copy paste it into a vector, and then we will amplify the plasmid using E. coli. So in this part I would like to walk you through the second part. So then we start first with the transformation of Agrobacterium tumefaciens, then we start with the transformation of the plants using this Agrobacterium tumefaciens, and then we will go through the selection of the transformed plants. So what is Agrobacterium tumefaciens? Agrobacterium tumefaciens is a bacteria that can create tumors in plants. Now, the tumors are a little bit different in the case that we would see in humans or in animals, but we do see some abnormal cell growth. And this happens due to the fact that these bacteria copy in specific genes encoding auxin and cytokinin. And these are plant hormones inducing cell growth. And it copies in one other gene, which is encoding a few opines, and this is for the nutrition. So basically it can use these opines as a carbon source and as a nitrogen source, and it grows very happy on this. So it's actually very smart that it also copies in these plant hormones, because that way you make sure that the plant is growing very nice and you have a lot of cells also producing food for you. So more cells producing more food for you, and therefore you also grow better. Now, however, these bacteria have only a specific part that they normally copy paste into the genome of a plant, and that is a part between the RBT-DNA repeat and the LBT-DNA repeat. So this whole part will be copied inside the plant genome, randomly somewhere inside the plant genome, but it doesn't matter what's in between this part. So normally here we find the genes for cytokinins and for the auxins and for the opines. But if we replace this part with, for example, in this case, a kanamycin selection with a promoter and a terminator, and our gene of interest, in this case LECSR, with a promoter and a terminator, it actually can also copy this whole part in there, and actually it copies in the gene that we want, and is very efficient in that. Now, first of all, we need to get this plasmid into the Agrobacterium tumefaciens. And what we do, so here we have a nice cell suspension of these Agrobacterium tumefaciens in a special cuvette with some metal plates here, and we add some of this plasmid into the cuvette, and we apply an electric shock. Some of these plasmids will enter into the cell, and some of them will not take up, but later we will select the the cells that are taking up the plasmid by using another resistance cassette, in this case also kanamycin. So only the bacteria that accept this plasmid have this kanamycin resistance and will survive on the kanamycin antibiotic. And then we will start with the transformation of the plants using this Agrobacterium tumefaciens. So as I explained before, here we have the RBT DNA repeat, and here we have the LBT DNA repeat. So this whole part will be copied into the plant. And of course we only have the Agrobacterium tumefaciens, which have taken up our plasmid. And we then use the floral dip method. We will use the floral dip method. This is a method that is mostly used for Arabidopsis thaliana, and what we do is we take the plant and we dip it in a solution of Agrobacterium tumefaciens. Then we will let the plants grow a little bit, and later we will harvest the seeds. These seeds we will put on the selection, and as you can see here, so here we have a plate full of kanamycin, and on some of these, some plants will survive. So here we see one, and there we also see one plant surviving. These are the plants we pick, and we transfer them to the soil, where we later will perform some other screening measurements. And in this case, we just look at MPQ, and we look at protein screening, but you can also do, for example, DNA screening or any other screening that is necessary to identify if your plants have taken up the gene of interest. Now, what's interesting to note is that we have a lot of these seeds on the soil. So I said that Agrobacterium is quite efficient, and it is. However, only 1 to 2% of the seeds actually take up the DNA, are actually transformed. Luckily for Arabidopsis, this is not a problem, because the seeds are very small and they produce a lot of seeds in every plant. So generally we get enough. So this is for Arabidopsis a very, very nice and simple strategy. In some other plants we cannot use this, but basically it always comes down to, if we want to transform a plant, we use Agrobacterium tumefaciens, and in this case we use it on the flowers, but sometimes we use it on leaf tissue, and we incubate the leaf tissue with Agrobacterium tumefaciens. We've looked at the transformation of Agrobacterium tumefaciens, the transformation of plants, and we've selected transformed plants. Now, as you probably are wondering, because I was very fast in this genotype, I was very fast in explaining this genotype, and this is a PSBS knockout mutant. Now, why it's a PSBS knockout mutant? Well, PSBS is responsible for non-photochemical quenching. If we knock out PSBS, and NADCSR1 is the only one responsible for quenching, we can more easily measure the quenching parameters only related to the NADCSR. But how did we even get this genotype? It is not a genotype that we normally find in nature, and that is because it doesn't have any highlight protection mechanism. And this is, of course, in nature, it's not a good thing, because you have the sun, you have very different types of light. So what we can see, here we look at the non-photochemical quenching curve of the wild type, and we see a huge curve of non-photochemical quenching if we apply highlight. However, in the MPQ4, basically we don't see any non-photochemical quenching, indicating that these plants are unable to perform that without the use of PSBS. So we have transformed the MPQ4 mutant, a mutant without PSBS, and we call this a PSBS knockout mutant. Now, how was this mutant made? And that's actually quite interesting, because this mutant was created by random mutagenesis. Basically what they did, they took the seeds of the Arabidopsis and they used fast neutron bombardment, or they incubated them with ethyl methane sulfonate. And this induces mutations in the DNA. Random, you cannot very specifically say I will induce a mutation here, but it induces mutations. So you get a lot of different plants from this, and you screen these plants until you find one that is interesting for your research. And in this case they found one which they identified as a PSBS mutant knockout. The disadvantages of this are the screening. You need a lot of plants before you find this one that is absent in this case non-photochemical quenching. For non-photochemical quenching, the measurements are actually quite easy. But if you would want, for example, where a mutant where the measurement is not so easy, it's impossible to create, because you don't need 4 plants as I've shown here, but you need thousands to millions of different lines to find one plant that is actually the interesting for you. So in some cases this is not an interesting method to create in order to create a knockout. Well, there's also another thing, and which is called a knockdown. And a knockdown mutant is not a complete knockout, so the gene is still there and the gene is still active. However, the total amount of protein is lower. So for example, I now want to have a specific mutant. This is an example also from literature, and they wanted an LHC2 knockout. Now, LHC2 is the most abundant chlorophyll binding protein on Earth, and it contains 3 genes. It's encoded by LHCb1, LHCb2, and LHCb3. However, these are not found in the library of mutants, and therefore they had to create these mutants themselves. So what they used was the interference RNA, and here we can see an overview. If we have the genome with a gene located on it, this is normally transcribed into messenger RNA, and this messenger RNA is translated. Now you know that this messenger RNA is single-stranded, and it has a sequence, for example, like this. And what we will do is we will use this interference RNA in the level of the messenger RNA. And this is also a system that has been used by plants and other organisms themselves to regulate the level of expression. So what you can do is you can insert a gene that is encoding a messenger RNA which is complementary to the messenger RNA of your gene, the interference RNA in the red. And this will be double-stranded. Now the cell does recognize the double-stranded messenger RNA and will immediately start breaking it down. So basically what happens is you have transcription, the messenger RNA is produced, but it's completely immediately degraded, and there will be no translation of your protein. Another method is the CRISPR-Cas system, and this is actually quite a novel method in the plant molecular genetics. What we have is we have the Cas9 protein, which uses a gRNA or a guide RNA, which partly binds into the protein, and it has a sequence that recognizes the sequence on the genome DNA, or on any gene DNA actually. And when it finds this sequence, it induces a double-stranded break. So here we can see the schematic. Here we have a Cas9 with the guide RNA in there, and what happens is that we get a double-stranded break. Now, of course, the cell can perfectly repair this, but the problem with double-stranded break is that it's not always easy to repair, and you will have some changes in your reparation system, and this induces mutations. Now, these mutations can therefore render the protein inactivated or make sure that the expression doesn't happen at all. There's one other interesting method. So of course you can use one single guide RNA that's targeting here, but what if we use also a guide RNA that's targeting here? Then we induce two double-stranded breaks, and of course the chances are higher that they will be repaired with mutations. Another thing that can happen, however, is that we completely lose this inner part, and that these are attached again. So now we have a complete loss of the gene, and we have a complete knockout. So these systems are actually quite interesting to use and will also be used in the near future. To summarize, so we've looked at how to create a mutant. We have looked at different possibilities of overexpression, making a knockout using, for example, random mutagenesis, but also using CRISPR-Cas. And we've also looked at a knockdown using interference RNA.